Good morning, children. Class 8. Today's topic is clotting of blood. Same chapter, circulatory system. Today's topic is clotting of blood. It means that how the blood can be clotted in our body. If suppose blood cannot clot in our body, then by a small cut, the bleeding when started, then it will continue till our full blood will not be secreted out, and afterwards we will die. So clotting is very must and very essential process done by our platelets. So children, first we should know about the platelets. Yet it is not in your book, but I am telling, please children listen it. Platelets. Platelets are the small or tiny particles of a mega or a big cell called as megakaryocyte. Here children, I am writing megakaryocyte. Megakaryocyte is a very giant cell found in our bone marrow, and this megakaryocytes, when broken into pieces, they can be changes into blood platelets. One megakaryocyte can give 1000 to 10,000 number of platelets at a time. Okay, now how the platelets help in our body to clot the blood. This is the main 4 steps which can help in the clotting. So this clotting always done by some enzymes. First of all, when there is cut or injury, so platelets will be there and injured tissue. Both these together they start to release one enzyme that is called as thromboplastin. At the place of injury, the injured tissue and the blood platelets from our body, that is blood, they will release one enzyme that is thromboplastin. This thromboplastin along with the calcium ion changes the prothrombin into thrombin. Children, one thing again I want to tell you, prothrombin, if this is also an enzyme, and if the pro in front of it, its name, and gen at the back of its name, if it is written, you can understand about it that this enzyme is releasing in an inactive form. Afterwards, in higher classes, you can study about it, why they are releasing in an inactive form. But if there is pro or at the last, just here it is gen, these are releasing in an inactive form. To make them active, see that prothrombin, they need calcium ion which are present in our blood, and thromboplastin where it is secreted just now. So thromboplastin and with calcium ion, this prothrombin changes into thrombin. This is the second step. Now come to the third step. This thrombin, or in presence of thrombin in our blood, there is one protein that is soluble protein, that means it is soluble in our blood, we can't make it out from the outside. This name is fibrinogen. See children, here it is also an enzyme and there is the gen word at the back. So this is also an inactive form of it. This fibrinogen It is a soluble protein found in our blood. When it releases in presence of thrombin, it becomes active and forms the fibrin. Forms the protein fibrin, and this protein is insoluble, so we can make it out. That means blood can be seen little bit dense due to the presence of this protein. As children, we have taken suppose water and sand. When sand will be settled down and water will come up, or suppose it is rotating inside the water only, then we can see water is not a very fluid. Water is somewhat muddy and it is having some more density. So here also, when fibrinogen, that is soluble protein in presence of thrombin, changes into fibrin, that is insoluble protein. Now this fibrin, when that third step is over, see, this is the first step, this is the second step, this is the third step, and this is the fourth step. So after third step, when there will be the formation of fibrin, that is insoluble protein, then in our blood there will be platelets. They release it, so there will be no platelet. Other cells are there, RBCs and WBCs. Along with that fibrin, they form a mesh or a network of fibrin, and along with that network, all the blood corpuscles are entangled into it. Due to that mesh made by the fibrin and along with the blood corpuscles, that is RBC and WBC, it get a mesh or reticulate structure or a network at that place, and slowly when it becomes dry, then it can form a clot. See children, after clotting, when you again snatch it or take it out, then again the blood started to come, because only on the upper surface there will be the clot. Inner surface or inner to it, there is no formation of new cells takes place. So it will take time. So that's why the clotting should be there in that area. Then till the new cell will not grow, it will not come up to the surface level, till then clotting will be there. Afterwards, clotting or that area or that hard part, solid part becomes removed by itself and new skin comes over to it. So these are the 4 steps taken in the clotting of blood. First, see children, I am repeating here. First, the injured tissue, and at the place of injury, the platelets will secrete one enzyme, that is thromboplastin. Thromboplastin along with the calcium ion, it changes prothrombin into thrombin. Prothrombin is inactive and thrombin is active form. Then this thrombin changes the fibrinogen. Again children, here it is gen. So it is also inactive form, but it is a soluble protein found in our blood. Then in presence of thrombin, fibrinogen again becomes active and gen will be removed. Only remains there is fibrin. Fibrin is the insoluble protein. And that insoluble protein, that is fibrin, along with the fibrin is a long thread-like structure in the protein as a protein. Then fibrin along with the RBCs and WBCs or along with the blood corpuscles form a mesh or a network, and then network when becomes dry, then it will form the clot. And this clot, till it will not dry, you should not have to remove it. And children, one thing more I want to tell you, bleeder's disease. It is known as hemophilia. Hemophilia is the disease where the blood cannot clot. And after one injury, they are releasing blood from their body and ultimately they die. This disease is known as hemophilia. Yes children, okay. God bless you. Thank you.